Part 2 of Chapter 3 is going to begin with ATP. So if you have your outline, this is Roman numeral 3, ATP. So ATP, you're looking at this molecule, which is going to be the energy molecule of the cell. Anytime we need energy for something to happen, a chemical reaction, an enzyme a channel protein to pump something across the membrane, we're using ATP. So when we eat food, the food that we eat has this potential energy, and our body can't use fats and it can't use carbohydrates directly. All of the energy stored in the glucose molecules or fats or amino acids or proteins, all of that energy gets used and harnessed to create ATP. So adenosine triphosphate is basically a modified nucleotide. So we have the A base here, adenine. So adenosine triphosphate, 1, 2, 3 phosphate bonds, and the most energy between the phosphates is between the last two. So the bond here is the most energetic bond. So when we cleave the phosphate group off of our triphosphate, we get ADP, adenosine diphosphate, right? So ATP, when you hydrolyze the phosphate off, you get ADP. But ADP, there's still energy. You can actually still harness the energy between the first two phosphate groups. When you do that, you get a molecule of AMP, monophosphate. That is the lowest energy molecule. You have to recycle that. Most of the time in a cell, you just put the phosphate group back onto ADP to make ATP. So there's two ways that you can put a phosphate group back onto ADP, and that is through substrate level phosphorylation, like you're looking at here, or it's with oxidative phosphorylation, which is this picture that happens on the electron transport chain. So let's talk about substrate level phosphorylation. So there's that word substrate. We know that we have an enzyme involved here. It's a very quick reaction. Our substrate, this is actually known as PEP. It's going to come in to the enzyme, and the enzyme will hug this phosphate donating substrate to ADP and transfer the phosphate group onto ADP to create your ATP. So it's done in the muscle cells a lot. This is something that can be done without oxygen, as you can see. Most of the ATP made in your body is not done through substrate level phosphorylation. It's done through oxidative phosphorylation, but this also is a way that you can make ATP. Just a interesting sort of supplement here. You can actually get this creatine phosphate. So creatine phosphate is, see if it's in the, so your muscle cells have creatine phosphate that donates a phosphate group to ADP. So this green substrate here is creatine phosphate in your muscle cells, right? So if you take in creatine phosphate, then you're going to have a lot more of these substrate. You're basically packing your muscle cells with this substrate so that your muscle cells are more able to make ATP in this other pathway, in this substrate level phosphorylation pathway. So bodybuilders will often take this supplement to hopefully give them more energy in their workouts, right? The oxidative phosphorylation is how we make ATP, the bulk of our ATP in our cells. And we're going to go through this process through cellular respiration, right? So this is the electron transport chain in cellular respiration. So let's get started looking at cellular respiration. So with cellular respiration, you know from the courses you've had before physiology that it's glucose. This is basically, we have glucose plus oxygen. The products of cellular respiration are carbon dioxide and water. So in this oxidizing of glucose, we are transferring the energy in the glucose molecule to create a molecule of ATP, right? And we do that by just adding that phosphate group back onto ADP. ATP then is used for work. So this is anything the cell requires energy for, to move, your muscle cells moving, transport of molecules across the membrane. We have pumps that we'll look at, and then creating things, so molecular synthesis, building molecules. So we're going to take a look at how glucose is oxidized and how we make our ATP. All right, so this is the first step. So you guys should remember this from your precursor class. 3 steps for cellular respiration: glycolysis, the Krebs cycle, and then the electron transport chain. You are looking at glycolysis here. So glycolysis literally means to split glucose. So the initial substrate we have is glucose. So glucose, there are 10, you can see the word number 10 here, 10 different chemical reactions that take place in glycolysis. And through the process of glycolysis, we are breaking down the glucose molecule, and then we are creating, at the end of it, we have created 2 molecules of pyruvate. So 2 molecules of pyruvate. Each pyruvate molecule is only 3 carbons in size. Glucose was C6, right? So we have our 6 carbon molecule that's broken down to 2 3 carbon molecule enzymes. So I just want to point out a couple of things here that will come back later on. The very first step of glycolysis is irreversible. So this enzyme, hexokinase, can take glucose and convert it to glucose 6-phosphate, but cannot do the opposite. Okay, it can't take glucose 6-phosphate and put it back into glucose. And then we have a rate limiting step here, this phosphofructokinase enzyme. That's the enzyme that controls the rate of glycolysis. So overall, what you see in these 10 different chemical reactions is that we are creating a little bit of ATP. We have 2 ATPs made there. We have 2 ATPs made here. But notice that we are using 2 ATPs also in the process. So we gain a net of 2 ATPs, right? So we made 4 ATPs, but we used 2. So we get a net of 2 ATPs. Another thing you want to notice about glycolysis is that we are transferring, there's a redox reaction going on, and we are transferring electrons to NAD. So NAD is known as the electron carrier molecule. It's going to take electrons away, so it's going to gain those electrons and get reduced, and carry those electrons to the last step, the electron transport chain. So we're creating a reduced electron carrier. All right. So going down the list in your outline, you are oxidizing glucose to pyruvic acid. You are making 2 pyruvate molecules. ATP is created, and the way that you're making ATP here is with substrate level phosphorylation. Okay, so substrate level phosphorylation, so using that enzyme to make ATP. It occurs in the cytoplasm of the cell, and it does not need any oxygen to occur, so this is an anaerobic process. It happens whether or not there's enough oxygen in the cell. All right, so this is just a schematic view of glycolysis. So we see that we get 2 NADHs, and then we're going to get 2 ATPs out of the process. All right, so which of the following is a result of glycolysis? And the answer is, right, 2 pyruvate molecules, 2 net ATPs, and 2 NADH molecules. All right. Now we need to talk about the second step, which is called the Krebs cycle. This Krebs cycle also has the other name, citric acid cycle, or CAC. And pyruvate from glycolysis, right, here's our pyruvate molecule, does not enter into the Krebs cycle directly. It actually changes form. So coenzyme A comes in and binds to pyruvate to create acetyl coenzyme A. Acetyl coenzyme A is going to go to the Krebs cycle. So coenzyme A, this was part of your vitamins. In the process of, so this is our enzyme that helps to catalyze this reaction. In the process, we are losing a carbon to carbon dioxide. So this is our waste product, one of the waste products one of the carbon dioxides that is, of course, our waste product of cellular respiration. And then we are also creating another electron carrier molecule so that we can bring it to that last electron transport chain for oxidative phosphorylation. Okay, so acetyl coenzyme A is the molecule that enters into the Krebs cycle. All right, so acetyl coenzyme A is our initial substrate. And for every acetyl coenzyme A, there is one GDP produced, which is the equivalent of ATP. So we have a little bit of ATP made, and then we have the bulk of our electron carriers, NADHs and FADH2. I like to call these Uber and Lyft. So they're both electron carriers. They're both going to carry electrons from glycolysis or from the Krebs cycle to the electron transport chain. They're just different molecules that do it. So NADH could be Uber, FADH2 could be Lyft. But this is really the main thrust. This is what the Krebs cycle can donate to the overall cellular respiration, is these electron carriers. Because again, we are taking the electrons away from glucose and we're going to give them to oxygen. All right, so oxygen is gaining those electrons. When it gains electrons, it's reduced. So we're reducing oxygen. We're oxidizing glucose, reducing oxygen. We are creating waste products. So the carbons that were in glucose are really not of importance because it's the electrons that we want to harness the energy from. We want the electrons to travel to the electron transport chain. So the carbons are lost as carbon dioxide. All right, so glucose, as glucose generates the acetyl coenzyme A molecules, this cycle turns twice for every glucose molecule. So this is just a quick look at the Krebs cycle. It occurs in the mitochondrial matrix. We can see that our first substrate is the acetyl coenzyme A, and then that creates citrate. So this citrate is also known as citric acid. And this is why it's called the citric acid cycle in some textbooks. Okay, but we can look around the cycle. We see that the last step, it joins with the initial substrate. So that's why it's a cycle. We are creating our NADHs, our electron carriers. Here's FADH2, an electron carrier. We're also at the same time losing carbon dioxides. And a little bit of ATP is made. All right, so this is a summary of the Krebs cycle, but again, the Krebs cycle really donates, the main benefit of the Krebs cycle is extraction of our electrons with our electron carrier molecules, bringing them to oxidative phosphorylation. All right, so this is our third step, electron transport chain. So the electron transport chain occurs on the mitochondrial inner membrane. So this area here, right, that's beige, this is the matrix of the mitochondria, the very inside. And then we have another space between the two membranes called the intermembrane space. So the outer membrane of the mitochondria would be up here, right? We'll just draw in the outer membrane. So what happens is the electron transport chain is a series of proteins. We call them complex 1, complex 3, complex 4. And then there are coenzyme Q and cytochrome C. Those are also carrier proteins that are in the electron transport chain. So the electron transport chain works like this, right? So we have our electron carrier molecule, which comes in, from the matrix. So this is the Krebs cycle is happening, making our NADHs and our FADH2s. We can also get shuttling in the NADH from glycolysis. All right, so what happens is the electrons are dropped off into the first complex. This protein, when it gets those electrons, it harnesses the potential energy in those electrons and it uses it to pump these little blue balls, which are hydrogen ions, H pluses, Now, H pluses are also called protons, okay? And the reason is a hydrogen atom is one proton, positive charge, and one electron, right? So this is a hydrogen atom. Hydrogen is a weirdo because it doesn't have a neutron. How do you get a positively charged hydrogen ion? Well, you take away the electron, right? If it loses the electron associated with it, then what's left? One proton. So that's why H plus, the H plus is the same as a proton, okay? So we have a lot of protons kind of in the matrix here, and every time the complex has those electrons, it will pump a proton from the matrix into the intermembrane space. Those electrons will get shuttled across with coenzyme Q to complex 3, where another proton is shuttled into the intermembrane space. Cytochrome C will then shuttle those electrons to complex 4. Same thing happens with complex 4. When it gains the electrons, it will shoot a proton into the intermembrane space. Okay? So what happens at the end is that we have our oxygens, right? This is why we breathe, why we die if we don't breathe oxygen. It's because oxygen is going to be the final electron acceptor. So the electrons that are finally in this complex 4 have nowhere to go because there's no other electron shuttling mechanism present. So it goes to oxygen. Oxygen is very electronegative. It has a high affinity for electrons, and it will grab those electrons away from complex 4. Those electrons will combine with oxygen, and it says half an O2 molecule, right? So we're just doing an O molecule. It's just one oxygen atom. But it combines together, and we have some protons that will be attracted to the process and create a molecule of water. So for every time, every time we have oxygen receiving, so oxygen is the final electron acceptor in this electron transport chain. When oxygen binds those electrons, it will form water. So this is our waste product, right? All right, so what did we do in this whole process? We have pumped protons across the inner mitochondrial membrane to create a very distinct concentration gradient, right? We have a high concentration of protons in the intermembrane space. We have a low concentration of protons in the matrix. And so through diffusion, right, we want to go from high concentration of protons, we want to diffuse to a lower concentration of protons. But protons are charged. They do not run through the plasma membrane because this is going to be a polar charge. It does not go through the lipid interior, which is nonpolar of the membrane. And so the only place where the hydrogen ions are allowed to diffuse down their concentration gradient is through this enzyme called ATP synthase. This enzyme has an opening in it. It allows the protons to go down their concentration gradient. I like to think of this as you're holding water. A dam holds water back. So this is, the hydrogen ions represent the water. And if you put a little hole in the dam, the water is going to run quickly through that hole, right? So there's a lot of potential energy stored in this concentration gradient, right? It takes a work to create a high concentration of protons. If you allow them to run down their concentration gradient, this is going to release that potential energy. The ATP synthase molecule harnesses the energy in this flow of protons so that it basically adds the phosphate group onto ADP to make ATP. Okay, so the ATP synthase enzyme directly gets its power to make ATP from this concentration gradient. Allowing the hydrogen ions to flow down their concentration gradient is going to make ATP. So the concentration gradient is paramountly important. You need that concentration gradient of protons, because if you didn't have that, right, imagine where you have a membrane, you have ATP synthase here, and there's just an even amount of protons on either side, right? is there going to be a driving force that allows these protons to move through the ATP synthase? No, right? There's no concentration gradient. Protons will not flow through in a meaningful way. And since the manufacture of ATP depends on the flow of protons down their concentration gradient, if you don't have a concentration gradient, you don't make ATP. So that's what these complexes of the electron transport chain work to do. They work to always create that concentration gradient. So throughout your whole life, you will always have a concentration gradient of protons different, right? In the intermembrane space, you have a lot of protons. In the matrix, you have low concentration. Let's look at cyanide. So cyanide is a poison, and it kills you very quickly. And it works on the electron transport chain. So cyanide is a, CN. It binds non-competitively to cytochrome oxidase or complex 4. So it binds here. It doesn't have to compete with anything to bind there. It just binds and latches on, and it stays on this complex 4. It alters the shape of its active site. So therefore, you see these arrows. These are the arrows that are where the electrons are going to get shuttled. But what happens is when the electrons reach this complex 4, if cyanide is present, it does not allow the electrons to be released to oxygen. So you can have your oxygen atom sitting right there, but the electrons cannot get past to oxygen. So what that does is it backs up the entire chain, the entire electron transport chain, because if these electrons don't move, then the electrons here are stuck. The electrons here are stuck. The electrons here are stuck. So you just get this traffic jam of electrons that cannot move forward. It's like standing in line and the last person in line is not going to move, right? So you can't go forward if that last person doesn't move. And so therefore what happens? Once all of the protons go down their concentration gradient, you are not building up that same concentration gradient, right? So you're going to allow the protons to reach equilibrium. The ATP synthase enzyme stops making ATP, and your chemical reactions will shut down, right? So the electron transport chain shuts down. ATP is no longer being produced. You're going to use up the cell's ATP in a few minutes, and so if you can't do any more chemical reactions, you're going to die. So that's how cyanide works. It disrupts the electron transport chain. Some terms that you have in the notes is proton motive force. So this is the potential energy in the concentration gradient. I talked about how, let's erase some of this word here. I talked about how the concentration gradient is super important, right, between the intermembrane space and the matrix so that ATP synthase can work because it harnesses the energy directly from the concentration gradient. And so this force, this potential energy in this gradient is called the proton motive force. Okay. That is how we make the bulk of our ATP in our cells with the electron transport chain. So looking at everything overall, here is the summary of basically the cellular respiration, glycolysis. We can see that we are producing a little bit of ATP, but also these electron carriers. In this initial step, the pyruvate to acetyl coenzyme A, we also have a little bit of electron carrier. The Krebs cycle making the bulk of our electron carriers. The electron carriers bringing their electrons to the electron transport chain where we create ATP using oxidative phosphorylation. So oxygen is used here to phosphorylate ADP. Okay, so oxygen is used at this last step. We can recycle our electron carriers so they can go back to the glycolysis and the Krebs cycle to pick up more electrons. The waste product of water is being made. Okay, so overall, for every glucose molecule, okay, we are going to create 32 ATPs. So for every glucose that we can break down in the cells, we get 32 ATP molecules. Pretty good. All right. What about if you run out of oxygen? So for most of your body cells, you cannot function without oxygen, but for your skeletal muscles can actually continue to produce ATP even if the oxygen's not there. So glucose catabolism, so breaking down glucose in the absence of oxygen. If there's low oxygen availability, so if you're running a marathon, if you're straining yourself, doing a very rigorous exercise, a HIIT program, for example, what can happen is the electron transport chain backs up, just like cyanide, right? You just don't have oxygen there, and the Krebs cycle stops. However, glycolysis can continue, right? That very first step that happens in the cytoplasm, it doesn't need oxygen present to happen, so it doesn't really know if there's oxygen there, it just continues to happen. And but as a byproduct, if you're not moving through the Krebs cycle, then this organic byproduct is created called lactate, or lactic acid is made. So let's take a look at that process, right? So glycolysis is happening inside your cytoplasm. So glycolysis is going to provide the cells a little bit of energy, 2 ATPs, to keep going, so your muscles can keep running, or you can keep doing squats or whatever it is for a limited amount of time, right? But as a byproduct, you're going to create lactic acid. So this is, so this is an acid, and the more of this that accumulates inside your cells, your cellular interior pH is going to drop, right? The pH, if you're making acids inside of a skeletal muscle cell, the pH is going to drop, become acidic, and then your enzymes will start to get affected, right? Your proteins and enzymes, they're going to respond to a different pH, and they're not going to work as well in a different pH. So you can only sustain anaerobic respiration for a short amount of time before you fatigue, and you're not going to be able to contract those muscles anymore. So, we're moving on to a separate subject. Anyways, so this is your nutshell fermentation. Your muscle cells are able to do this. And they also think that when you're sore the next day, this is because the lactic acid built up inside of your muscle cells, and this is going to cause that pain of soreness. The lactic acid, by the way, does move into the bloodstream, and then your liver can metabolize it back to pyruvate. So we can do this opposite reaction. So lactic acid can be removed from the body. All right, so that's the end of part 2.